morning, I am Madhurima Datta, Zoology and Biology Educational Content Creator. Now's content for B.Sc Zoology Honours, Microbiology Honours, Physiology Honours and Biochemistry Honours. And also for exams CUET PG, GATE, DBT exam, IIT JAM exam, CSNET Life Sciences exam and M.SC in Zoology. Unit 1, Introduction of Genetics, Paper Genetics or Inheritance Biology. And topic is allele and locus. Definition and concept of allele or allelomorph. An alternative version or functional state of a gene occupying a specific position or locus on a homologous chromosome is called allele. Origin: this allele term coined by William Bateson in 1902. Molecular basis of the allele. It is arise due to variation in the nucleotide sequence of a gene locus via point mutation, insertion, deletion or substitution. Next, types of alleles. Based on phenotypic expression and prevalence, these are wild type allele. What is the wild type allele? The most common naturally occurring allele in a wild population, usually encoding a fully functional gene product. It is a capital symbol or plus symbol. Next type is mutant allele, a modified form of the wild type allele resulting from DNA mutation leading to altered, reduced or lost gene functions. Next, null or amorphic allele causes complete loss of function of the gene product. Next, hypomorphic allele result in reduced expression or partial loss of function. Next, hypermorphic allele result in increased level of normal gene function or overproduction of product. Next, neomorphic allele grant a novel gene function or altered expression of patterns. Next, historical evolution of the concept. Mendelian unit factor, a discrete unit of inheritance controlling a specific phenotype trait. Next, classical view. Pre-1950s, the gene was viewed as an indivisible unit of functions, mutations and recombinations. Next, Banger-Finn structure analysis. Seymour-Wenger's genetic mapping of the R2 locus in bacteriophage T4 disproved the classical model, establishing three distinct operational units. First is cistron, the functional unit defined by the cistrons complementation test, which is equivalent to a modern gene coding sequence. Next, muton, the smallest element of DNA that when altered causes a mutation, a single nucleotide pair. Next, recon, the smallest elements capable of undergoing genetic recombination, that is a single internucleotide bond. Functional classification of alleles. H.J. Mullers in 1932 categorized alleles based on their functional behavior relative to the wild type allele. This is a plus denotation, gene product and dosage responsiveness. First type is amorph or null allele. Functional effect, completely loss of function. Gene product, zero functional transcription or protein product. Example, nonsense mutation, whole gene deletions, frame shifts leading to nonsense mediation decay. Next, hypomorph type. Functional effect, partial loss of function, reduced expression levels or reduced specific activity of the protein. Example, promoter or enhancer point mutation, missense mutation, reducing catalytic efficiency. Next, hypermorph types. This is a gain or normal functions effect. Gene product, it is an increased protein level or elevated catalytic activity. Example, promoter duplication, constitutive promoter activation mutation. Next, antimorph or negative molar morph type. Functional effect, active interference, gene product phenotype, mutant product poisons or disrupts the activity of the wild type product in the same cells. Example or mechanism, common in multimeric or structural proteins. Next, neomorph type. Functional effect, gain novel function, protein acquires a new enzymatic activity or is expressed in an ectopic spatial temporal domain. Example, DH1 by 2 mutation in glioblastoma producing 2-hydroxyglutarate or BCR-BL1 fusion protein. Next type is isomorph type. Functional effect, identical function. Gene product or phenotype, identical phenotype produced across variation in gene dose. Example or mechanism, it is a silent or synonyms mutation with identical expression levels. AP alleles and advanced regulatory dynamics. AP alleles are genetically identical sequences displaying different heritable phenotypic states due to mitotic or meiotic inheritance of epigenetic modification. Epigenetic modification is DNA cytosine methylation, histone marks, etc. This will be known in future slides. Next, obligate AP alleles. It is strictly tied to underlying DNA sequences variants. Next, pure AP alleles. Heritably methylation alteration occurring independently of any genomic sequence alteration. The topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima for all video of graduation and masters, and follow my Facebook account Madhurima Datta. Thank you everyone.